In this video, we'll be looking at some ostriches, and also we'll be looking at prokaryotic cells. So we know there are prokaryotic cells and eukaryotic cells. This image right here is an example of a prokaryotic cell. Bear in mind that when I talk about a cell, I don't talk about the one you see in your book that is like flat. All cells are actually 3D. You have to bear this in mind. So the image that you see in your textbook is a picture of a cell that has been cut open so that you can see the inside of the cell. But in reality, all cells are 3D and actually they're closed. So you can't see what's going on inside unless you cut it open, like in this image. Okay? So you can see a prokaryotic cell is quite unique. It's hairy, it's got this long tail. It's pretty different from a eukaryotic cell. So that's the purpose of this video. So first I want you to try and label this picture of this prokaryotic cell and see how far you can get. How much things can you label? Now first, what does prokaryotic mean? Okay, pro means before or prior to, whereas karyotic means nucleus. So before nucleus. What this basically means is that prokaryotic cells do not have nucleuses. They are cells that don't have a nucleus. They are before nucleus. Doesn't make that much sense, but you gotta remember they are before nucleus, meaning they don't have a nucleus. They still have DNA, this is the DNA, but they don't have a nucleus. There's no membrane surrounding their DNA, no nucleus. Now, when I was learning about this, I was like, God damn, why do I care about prokaryotic cells? Like, where can I even find them and why do I care about them? Because shouldn't we only care about eukaryotic cells because humans and plants and animals are made up of eukaryotic cells, not prokaryotic cells? So why should we care about prokaryotic cells? So prokaryotic cells are bacteria. The biggest example is bacteria. Bacteria are prokaryotic cells, okay? Humans and all that kind of thing, plants, animals, all that is made up of eukaryotic cells. Okay, so that will be another video. So why do we care about prokaryotic cells? We know prokaryotic cells are things like bacteria. So why do we care in the first place? We know that bacteria can infect us and give us disease, right? Depending which type of bacteria infects you, you can get a different disease. So knowing about the structure of bacteria and knowing all the parts that make prokaryotic cells, we can develop medicine to specifically target those parts of the cell. And that way we can kill the bacteria that infected us and become healthy. So that's why we have different medicines. When we eat that medicine, that antibiotic will have a function to destroy a certain part of the cell so that the bacteria can die and we can get healthy. So the main reason we care about bacteria and learning about prokaryotic cells is because at the end of the day, the doctors want to learn how to kill them so that we can live healthily and get rid of all the bacteria we have. Okay, because for example, we can make a medicine that can destroy this structure. This structure is called the flagella and it makes the bacteria move. If we destroy this, then the bacteria can't move around you anymore. Okay, do you see what I mean? So depending on the structure, you can develop a different medicine and so destroy the bacteria and be healthy. So that's why we care about prokaryotic cells at the end of the day. So here I labeled it. So first, you can see there's a whole bunch of names. So it may initially seem like, God damn, there's so many. How am I going to know this? It's not that bad. So I'm going to go over one by one and hopefully it makes sense to you after I explain it. So let's start off with this one first, the flagellum. This is basically a machine, a tail that moves. So not all prokaryotic cells have it, only some do. So not all of them need it, but some have it. And it's going to make sure that this bacteria or this prokaryotic cell can move around in whatever person or thing it infected, okay? But not all of them have it. So it's for movement. Now, the next one I want to talk about is cytoplasm. What is cytoplasm? Cytoplasm is pretty much the jelly-like or liquid solid. It's like a mix between a liquid and a solid. And it is inside the cell, okay? It is basically, I'll show you this picture again. It's this jelly-like substance inside the prokaryotic cell. It's basically like water, okay? So it is the material in which everything inside the cell is dissolved in. So the DNA is in it, there's ribosomes in it, there's things like all of those kind of things are in it. So the cytoplasm is pretty much just a liquid in which all the other things are dissolved in. Okay? Next, and it's important, the cytoplasm, because it's also got some enzymes in it. And if you don't know what an enzyme is something that makes reactions happen. It's like a protein that helps speed up reactions. So inside the cytoplasm, there are some enzymes, which we can't see in this image, but they can help speed up reactions that the cell needs to do in order to survive, like reactions to make energy and all this kind of things. Okay. The next one we should talk about is ribosomes. Ribosomes are basically these small dots. Okay. Ribosomes is a place where protein is made. A ribosome is a place where protein is made, because we know that proteins can do many different kinds of functions depending on the type of protein that we're talking about. Some proteins can make the cell wall, some proteins can help make energy, some proteins can help pump waste out of the cell, some can help bring good things into the cell. So we have proteins have many different kinds of functions, and we have many types of proteins, even proteins that help make up this flagella to move. So all those proteins are made in these small dots called the ribosomes. Okay, and specifically in prokaryotic cells, we have something called 70S ribosomes. This is pretty much just a unit of measurements of the size of a ribosome, essentially. So prokaryotic cells have 70S ribosomes, whereas eukaryotic cells have 80S ribosomes, so kind of slightly bigger ribosomes. Okay, so just remember that for prokaryotic cells, it's called 70S, not 80S, 70S. Now let's go on to what these two are, nucleoid and plasmid. So nucleoid is the DNA, the chromosomes inside the prokaryotic cell. You can see it's really a mess. It's like a blob. There's no membrane surrounding it. So in other words, there's no nucleus, like I just mentioned, because in the eukaryotic cells, so the cells that humans have, we have a membrane, a nucleus, and inside that nucleus we have this blob. But prokaryotic cells don't have that. So we call it naked DNA. Naked meaning there's nothing covering it up. There's no house for the DNA, like in eukaryotic cells. It's just naked, right there in the cytoplasm, all by itself. It's not protected by a membrane or anything like that. So that's why it's called naked DNA. We know the purpose of DNA is to code for proteins, so that proteins can be made. It's basically your code. It's the code for that cell. Okay, so it's basically how can I call this, the manual of how the cell works. So if the manual is here, you can read the manual and make all the proteins you need, and the cell can work properly. Okay, plasmid is essentially also DNA. It's not part of the main DNA. So this is the main DNA called the nucleoid. Here we have a little bit of DNA called the plasmid, but it's not part of the main DNA. And the plasmid's function is also to make proteins, or to be the script, or to be the code for the proteins. But specifically, you can actually prokaryotic cells can move this plasmid to other prokaryotic cells, because this plasmid can contain DNA for survival. It can contain a code for how to survive against a specific organism, and it can send it to other cells. So it's a special kind of DNA that can be moved in between bacteria to help them survive. Because, for example, say we get infected by a bacteria and we start eating antibiotics. Maybe we will be able to kill 99% of the bacteria that way. And there's this 1% of the bacteria, or 1% of the prokaryotic cells, that are immune to that antibiotics. And the reason why they are immune to that antibiotics is because of this plasmid. They have a gene that can help protect them from it. So now they can be like, oh, all my friends are dying, so I need to help them. And what they do then is they send this plasmid, a copy of this plasmid, to their friends, and now their friends are also immune. And that's why after a while we need to start using different antibiotics. Otherwise, all the prokaryotics become immune to it, and you won't be able to get rid of them. Okay, here we have pili. So pili have many functions. It's basically a shorter version of the flagella, and its purpose is to attach to surfaces. So it's like little hands. It can attach to other cells. It can also transfer these plasmids I just mentioned. The plasmids are transferred through the pili to other cells. Okay, so they're also useful for transfer of plasmids, but mainly attachment and communication. It's used to communicate and attach to other cells. Okay, cell wall. Cell wall is this layer in the middle. It is useful for structure. So it keeps the cell strong and prevents it from bursting. Because sometimes a cell can be harmed, and if the cell wall is there, it can help it stay intact and not burst. How about this one, plasma membrane? The plasma membrane is the inner layer. It has a very important function. The plasma membrane is the layer controlling what goes in and what goes out of the cell. So it can be like, okay, no, I don't want that thing coming into the cell, so I'm not going to let it. Or I don't want this thing to leave the cell, so I'm not going to let it. So it has control of what leaves the cell and what comes into the cell. So it's kind of the transport controller. It controls all of that all the kinds of movements of things into and out of the cell. Now the last one, so plasma membrane is not that useful for strength. It's not like the cell wall that is useful for strength. It's mainly for controlling movement. Okay, the last one here is capsule. Capsule has a similar function to cell wall. It's for strength, but it's also useful because the capsule is slippery. So sometimes it can help the bacteria survive inside our body, because we have many kinds of cells in our body that are supposed to get rid of these bad things, these bacteria or other prokaryotic cells that we don't want. So because sometimes, not all of them, some of them have capsules, not all of them, just like the flagella, not all of them have flagella. So just like that, not all of them have capsules, but some of them do, and it makes it very difficult for our body to grab them and get rid of them. Okay, so it's very useful in protecting the bacteria from getting eaten by our cells. So I hope all of these definitions made some sense. Here I have a summary of all the ones we just talked about. So I put it in word form in case you missed something. But if you understand all of these, it should that's all you need to know about prokaryotic cells.